So thanks for the organizers for inviting me. So what I'll basically be discussing in this talk is a lot of results dealing with this paper that was published a few months ago in JCIM. Um, yeah, so I'll get started. Maybe it goes. Okay. So first, maybe I should talk about what a protein ensemble is for people who don't do lots of MD simulations of proteins like I do. So a protein is a one-dimensional sequence of n amino acids embedded in a six n-dimensional space. And so basically, what people do is there's some sequence of a protein. Somehow people express it, bacteria or something, and then you can throw it into a synchrotron, and then you get out an X-ray crystallography structure. So you get one structure. And then what people proposed a long time ago is that you go, once you have the structure, you go from the sequence to the structure to the function. So you get the sequence from the DNA. The DNA is expressed into these amino acids to make a protein. The protein folds on a ribosome or somewhere else in the cell to form a specific structure that's resolved in these X-ray crystallography experiments. And then these proteins have a specific function that they carry out in the cell that does some physiologically relevant thing. So the example protein I've shown here is HV protease dimer. Um, Basically, this HV protease, what it does is HIV is a retrovirus, and it expresses when HIV infects a cell, it expresses its genome. It basically hijacks the cell's replication machinery. It uses reverse transcriptase to go from RNA to DNA, and then the DNA is expressed by the cell. But then all the proteins that's encoded in HIV's genome, it's expressed as a single long protein, and this HIV protease basically is a aspartyl protease that cleaves this what's called the GAG polyprotein into all the functional elements of the HIV proteome. And so basically there's a catalytic site here and it breaks peptide bonds using this catalytic mechanism. And so that was sort of like, gospel for a while in structural biology. And then people actually figured out that proteins move around and do stuff because you have these different states. And so for HIV protease, this matters because these flap loops up here, they can open and close, and whether they're open and closed basically allows substrate to enter into this active site and bind to it. So this H3 protease is sort of, interesting for from a pharmaceutical perspective and a biological perspective, obviously, because it's necessary for HIV to be infective, but also it mutates a lot. Um, so this protein mutates a lot, and these mutations sort of change the dynamics sometimes significantly. And whether these loops open and close at different rates or with in some characteristic way, it sort of determines whether inhibitory drugs, can bind to the active site or not. But so the point is that people figured out for this protein and lots of other proteins, or lots of examples of this at this point, was that you actually need to understand what the ensemble is doing. So you need to get not just a static structure, you need to get a bunch of different structures. And then this sort of ensemble of structures encodes the dynamics of the protein. So in this case, whether the flaps open and close, allowing substrate to enter the active site or not. And then it's actually the dynamics, not just the structure, that encodes the function of the protein. And so from a statistical physics standpoint, what we really want to be able to sample and calculate is like this partition function, right? So the partition function is just this integral of the Boltzmann factor over phase space. Generally, the energy of a system, U of PQ, doesn't depend on the momenta P, it just depends on the coordinates Q. So we sort of get rid of the integral over P, and we're just really concerned with this configuration integral. And if we can calculate this partition function, which the relation between a protein ensemble and this partition function is that, the ensemble of the protein is basically, all values of Q it can sample. Um, and then, so we want to calculate these Boltzmann factors. And if we have the entire ensemble of the protein and we know the energy function, we can get the ensemble. Or if you have experiments that somehow are probing the ensemble, you can in theory, get an energy function out of that. But the point is that people want to calculate and resolve these protein ensembles, which is in a lot of ways equivalent to just actually calculating the partition function. And once you have the partition function, you can calculate these observables using the Boltzmann weights and the partition function. And for example, you can calculate delta G of folding or delta delta G, or the change in delta G as you introduce a mutation or something like that, or you change the environmental conditions or something like the radius of duration, some structural properties of the protein. But the issue with this is that unless you're dealing with like an Ising model or a lattice gas or something, or an ideal gas, partition functions are hard to calculate. So typically the partition function for, a protein is highly non-analytical, and we need a way to estimate it, or, somehow estimate what this partition function or the Boltzmann weights are, or what the ensemble looks like. And so for, however many years, I forget when MD was developed, like for proteins, like in the 70s maybe. So for like the past 50 years or something, we've had these Monte Carlo molecular dynamics methods where, if you're not familiar with it, what molecular dynamics basically does is you approximate the energy function as a sum of these terms. So a van der Waals term, an angular term, a dihedral term, a spring constant term between the bonds and the protein, and then an electrostatic term. And sort of you have a bunch of what are called force fields that approximate these energy functionals. And then now we have an energy. So the force is the negative gradient of the energy. So once we have forces, and then we have a starting position from, say, a crystal structure deposited in the protein data bank, now we can use this energy function. We can calculate the forces. We have an initial condition. We calculate the forces and update the positions and velocities, normally using, at this point, GPUs on supercomputers. And then what these molecular dynamics simulation software outputs is a time series of the protein, basically wiggling and jiggling in water or some sort of solvent. And this is an approximation of our ensemble. So the whole point of this is that we want these protein ensembles, and a way to sort of approximate them is using these MD force fields. But now recently, in the past, I don't know, 5 years or so, there are these things like BioEmu and Boltz and these diffusion model frameworks, and then AlphaFold 2, where you can jitter and reduce the MSA. And you have these other machine learning approaches that use diffusion models or transformer architectures or a combination of the two, to basically, instead of, they don't really have an energy function. What they do instead is they take a bunch of structures from the PDB and MD simulations, and they train a bunch of weights and minimize the loss function. Okay, so isn't an exact analogy, but the point is with these ML models, instead of having this analytical energy function, you have this probabilistic loss function that's minimized. And then sort of the idea here is that you effectively learn sort of the pseudo energy via the loss function, I would say. So these weights of AlphaFold, um, I think conceptually, this is at least my opinion, that they can be thought of as sort of learning an energy function because they're sort of designed to calculate crystal structures, and crystal structures are supposed to be the lowest energy states of these proteins because they're being crystallized. But instead of doing MD, what these things do is you put a sequence, you give them a sequence, and then there's a neural network, and then they give you an ensemble of protein structures. And sort of an issue with these is that we don't really know a priori the quality of the ensemble or of the static structures that are put out. So things like AlphaFold have these metrics like PLDDT or PAE or the TM score, and they're something that can tell you, with reference to a crystal structure, how good the approximation is. And at this point, it's well known that here I'm showing some examples from this review paper from last year where um, there's a prediction of the structure from AlphaFold, and then here's the PDB structure, and it's sort of not doing very well at predicting these tertiary structures. So AlphaFold 2, its native implementation, it just goes from sequence to one structure. So it's approximating these like X-ray crystallography experiments I showed at the beginning. But like I said, people are normally interested in approximating the protein ensemble because we know you need to know something about the ensemble to calculate dynamics and then say something about the function of the protein. So recently, there are these mostly diffusion model techniques that instead of just outputting a single structure, they output an ensemble of structures. But in it, these methods have the same issue as AlphaFold 2 in that the structures they output, they may or may not represent physical reality to, a satisfying degree. So, for example, these are figures pulled from the SI of the BioEmu paper in Science. And here on the left-hand side, this is this validation set of proteins that have less than a 60% sequence similarity with their training set, or at least the AlphaFold training set, I think. And these plots are basically showing their heat maps. I didn't put the scale, but the qualitatively is we only need a qualitative understanding of these. But it's these root mean square deviations from one of these two reference crystal structures that are shown here in this left-hand column. And the point here is that for a lot of them, mostly the regions they're sampling are very far away from the crystal structures, which may or may not be okay depending on the specific structures that are being output. So one issue that these methods have, and sometimes they automatically filter them out, is that the structures being output don't satisfy like these steric constraints. So you have very unphysical structures where they have bad bond angles or bond links, or just the atoms are overlapping, which is kind of a no-no. So the point of what I'm going to talk about here, if it'll switch slides, is that it's from a review paper from last year. We need to add physics, more explicit physics to these models to, make the outputs more trustworthy. And I think this is something that's sort of general in the ML community at this point, especially in biology and chemistry and physics. And so one way we can do that is you have these outputs from the neural network, and then in this case, it says being used to parameterize an MD engine. So this is what we're going to use this is what we used in the paper that I'll talk about shortly. But the point is that we can take these output from the neural networks, and I said the title of the talk is, it has the phrase Langevin prior in it. So what we're going to do is we're going to take these structures output from like BioEmu or AlphaFold 2. We're going to use it to parameterize a potential mean force from a Langevin equation, and then we'll sort of compare the ensembles that are output, the time scales, and the fluctuations predicted from the ensemble, and see how they compare to benchmark MD simulations. Um, so ideally, we would compare them directly to experiments, but this was sort of like the beginning of this research project. And we sort of just wanted a crude way to apply approximate timescales to these ML ensembles because there's no integrator inside of these neural networks. So each structure, we don't know we don't know the time step between them. It's more like Monte Carlo, and we wanted to approximate, timescales of these ensembles that are being output by the neural networks. And so this is how we put some physics inside of it, is we couple these outputs to a Langevin equation, and then we can get some timescales and fluctuations and things like that of the dynamics. So how this workflow is going to work is that we use a neural network to output these structures. We do a projection operator to project these structures in this delta Ri basis set, which is the deviation of, it's a little bit hard to see, but there's sort of like a translucent structure here that's supposed to represent the average structure. So it'd be like the crystal structure. But then each of these structures has a deviation away from that. So our basis set for the analysis will be this delta Ri basis set, which is the fluctuation of residue I away from its average structure. And then we'll do a transformation into these Langevin mode spaces, which here I'm plotting a free energy surface, and the free energy is given by the contours in terms of the first two Langevin modes. And these modes each have a characteristic time and length scale, and they're independent of each other. And so what we can do is we can plot the structures. So here we're projecting the structures output from an ML method, and we're projecting them onto this 2D space. And the ones that actually end up in regions that are sampled in microsecond MD simulations, they're colored blue. And then the ones that end up in regions that aren't sampled in the MD are colored gray. And so what we can do with this approach is we can sort of filter out what we would call in this case, these are like deep fake structures with reference to a 1 microsecond MD simulation. So in this case, we can sort of prune out some structures that we say are unphysical on the one microsecond timescale with respect to our force field. So this is not, necessarily an exact approach in terms of saying, oh, these are just like total hallucinations by the model. They're hallucinations just in our frame of reference, in this 1 microsecond frame of reference, and with respect to this force field. But so what the Langevin equation allows us to get out is now we can sort of, if we make the approximation that the dynamics are Langevin, then we can calculate time correlation functions. And what happens is that it's probably not a surprise because we're introducing, new prior information. But if we filter out these structures and we don't use them, if we filter out these, out of set structures from the calculation of the potential mean force of this Langevin equation, and then we go back and we calculate these time correlation functions, the top plot is if we keep all structures, the bottom plot is if we remove these structures that fall outside of the span of the 1 microsecond simulation, then we can get time correlation functions that are generally in better agreement with OMD. And the colors here refer to the MSA depth used in AlphaFold 2. And I think I'll show this later, so I can go into more detail in a little bit. Um, but so this Langevin equation for protein dynamics, what we're doing is we're taking the statistics from the ensemble, and this is the case if we calculate it from MD simulations. So in MD, we have a crystal structure, we solvate it, we do the simulation, and then we coarse grain it, and then we calculate this H and A matrix, which this H matrix is a hydrodynamic interaction matrix. The A matrix is the potential mean force that's calculated just from the zero time lag covariance matrix of either from our protein ensemble. So we calculate the covariance over the protein ensemble, say output from bioemu. And the details, I'm not going to go too into the details of this method because they're in this paper at the bottom. And if you want the details, you can read it. But basically, what this approach allows us to do is we can calculate this hydrodynamic interaction and potential mean force matrix. And these sort of are input physics in the model, okay? Because the hydrodynamics tells us how the residues, how their motions are coupled via the hydrodynamic continuum represented by the solvent and say an MD simulation. The potential mean force is calculated based on the covariance of the residues with respect to each other. And that tells us that's a proxy for how strongly the residues are interacting with each other. So it's like a bead spring model, but nonlinear. So the residues can talk to each other from a long distance in sequence, and then we get the time correlation functions that I showed earlier. And if we diagonalize this equation of motion because it's coupled, there are two summations here, we can get a Langevin equation of motion just in terms of these modes that are all independent of each other. And they decay with a time scale that's the sigma lambda A, where the lambda A are the eigenvalues of the HA matrix, and the sigma is 3 kBt over gamma, I think. And that's sort of like a characteristic relaxation timescale. So when you do this, we get time correlation functions, so we get kinetics, and then we also get dynamics coming out, like things like RMSF. So what we did in this paper was we have results for HV protease dimer, which is what I've been showing earlier, and it's the functional form of the protease. But we also looked at we did a lot of variant effect prediction, not variant effect prediction, but the effects, basically mutation effects on the dynamics for this HV protease monomer. So we looked at 6 variants of the HV protease, and here I'm showing results for 2 of them. And basically what I'm showing in these plots is the root mean squared fluctuation as a function of, method. So if we're using AlphaFold 2 with this MSA depth of 8, which is a fairly low MSA depth, it's the lowest one we tested. If we use bioemu, or if we use bolts, which is another diffusion model for generating protein ensembles. And then black is our benchmark 1 microsecond OMD. Um, so one thing is that the sequence matters for these approaches. For MD obviously matters, but then also these ML methods have some knowledge of the fact that there's mutations. And the question is sort of that we didn't answer too much in this paper, is how correct is it getting the mutational effects? I think one thing that I at least expected to see that we didn't end up seeing in the final results was I kind of expected like a better trend of things like the RMSFs and the predicted relaxation timescales, which now I'm showing here in these top two plots. I would have expected that those had been some sort of function of the Hamming distance of the sequence away from the wild type, because what people have seen before is there's very, very different dynamics between the wild type and the mutants. And because these mutants are sort of loss of function mutants or gain of, forget, I don't know if I should call them loss of function or gain of function mutants, because the mutants are sort of evading the inhibitors. So their flaps open more, which I think sort of prevents it allows the inhibitors to leave more readily. But we didn't see that. But you can see from these plots just qualitatively. Um, Here, what I'm plotting is the relaxation time scale of each residue as a function of sequence predicted just from the theory. And I'm comparing it again, the same thing as in the bottom plots, but what we can see is that, at least in this case, is that BioEmu does a pretty good job for both of these sequences at approximating relaxation timescales. And that again, there's a sequence dependence to it. And then depending on which method you're using, if you're using AlphaFold 2 with a very low MSA depth, or BioEmu or bolts, you can get very, very different time relaxation timescales, which are sort of corresponding to how big is the span of this conformational ensemble. So depending on the method you use to generate your protein structures, you can get several orders of magnitude difference in the time scales that these ensembles are spanning. Um, so what we wanted to do was sort of quantify this by looking at the time correlation functions predicted from the theory, which is the dashed lines. So the dashed lines are the time correlation functions predicted from this Langevin equation using the potential mean force that's parameterized by the ensemble's output from the Ilmo method generated. So MSA of this certain depth is AlphaFold 2 with that MSA depth. AFC is AlphaFold cluster. BioEmu is BioEmu. DIG is, what does that stand for? Distributional graph former, which I don't think people use too much because it's not very good at generating ensembles. The MD, don't worry about it. And then the solid lines are the sort of benchmark time correlation functions from the MD simulation. So what we did is this is just a qualitative plot sort of showing that, again, depending on which method you use and what time scale of MD you're looking at, that will change sort of these will change depending on the ensemble that you're looking at. So, for example, in the top one, I'm just showing three residues across the sequence of HEV protease of the monomer. For example, for this first residue, the time correlation function from the 1 microsecond MD is fairly well approximated by, for example, AlphaFold 2 with MSA depth of 8, right? So that sort of indicates that for residue 1, this ensemble that's generated by AlphaFold 2 at an MSA depth of 8, is quite good at approximating microsecond dynamics that are seen from MD. But so what we did was actually we had some quantitative ways to calculate the distance between these time correlation functions. And then we used that to make these plots, which are telling us the fraction of residues that are best explained by each generative ML technique. So in this slide, we're just looking at AlphaFold 2, and what we found was, like I said a few seconds ago, was that if we have an MSA depth of 8, that's best at explaining approximately 50% of the dynamics of 50% of the residues in the protein at the microsecond time scale. If we look at the 100 nanosecond time scale, it's fairly robust MSA depth. You can pick things between 8 and 128, and those are those are all reasonably good at describing the 100 nanosecond, the dynamics of the 100 nanosecond timescale that's seen in OMD of that length. And then if we look at OMD length 10 nanoseconds, that's well spanned by AlphaFold 2 if you use the native 512 MSA depth. Um, and then here I'm just showing some examples of if I project the structures of from I forget which this is AlphaFold 2 at some MSA depth. Just showing that for these different sequences of the HIV protease monomer, you sort of have different percentages of these predicted structures falling outside the span of the 1 microsecond simulation. Another way to quantify this is if we look at the relaxation timescales predicted from the AF2 ensemble versus the OMD at 1 microsecond. And generally the correlations are pretty good for all the 1 microsecond, 100 nanosecond, 10 nanosecond timescale for all 6 sequences that we studied. But generally, if we look at the slopes, if they were perfectly correlated, the slope would be 1. If we use MSA depth of 8, that's always the best at the 1000 nanosecond or microsecond time scale. And it's also very good at 100 nanosecond. And then as we get to 10 nanoseconds, you can see how the full MSA depth is generally, pretty good, as well as the 64 MSA depth. So that was AlphaFold 2, and if we use it for these other methods I talked about earlier, like BioEmu or AF2 cluster, here I'm just showing some movies, the structural ensemble output from the reference MD, from AF2 at MSA depth of 8, and then BioEmu, and AF2 cluster. This is just very, very qualitative. But one thing to note is that generally AF2 cluster, it's spanning a much longer time scale. Well, we don't know if it's spanning a much longer time scale. It's something we still have to look into than the 1 microsecond time scale, or if it's generating just a lot of these unfolded structures that may or may not be something that are realistically seen in experiments. But when we go back, when we look at the percentage explained for these other generative models, we find that BioEmu is also pretty good. It's almost as good as AlphaFold 2.8 at describing the dynamics of the microsecond timescale. And it's also the best of the 100 nanosecond timescale, whereas AF2, AlphaFold 2 with full MSA is still the best at approximating the protein ensemble at the 10 nanosecond timescale seen in OMD. And then here again, I'm just projecting all these structures onto the free energy surface from the microsecond OMD. And this is just showing again all the different methods, they predict very different structural ensembles. And we can do the same sort of quantitative thing as before. This DIG method is not very good. Only really outputs structures close to the crystal structure. But this is just recapitulating what's shown in the top plots that BioEmu is pretty good at the microsecond timescale. AlphaFold 2 cluster is also generally pretty good. But yeah. So the last thing I'll sort of talk about is that we can use this, as I said before, as a filtering method. So what we can do is we remove this yellow structures that aren't sampled in the microsecond OMD. If we compare the RMSF calculated using, the full output ensemble, which is the red, versus the blue, which is corresponding just to the blue points here, we greatly reduce the observed fluctuations in the ensemble. And also we see a reduction in the timescales, which is, perhaps not surprising. But the point of this is that, for these residues where it more closely matches the RMSF, we can see that if we plot time correlation functions calculated from the ensemble versus the reweighted ensemble, I'm cherry picking a little bit here, but we have the potential to much better match the time correlation functions seen in the simulation, which I showed earlier, and it shouldn't be that surprising because we're incorporating more information that is simulation specific. Also works for dimers. I already showed this at the beginning. I'm sort of running out of time, I think. But the dimer, it's something where the reviewer asked us to do it, and we should have done it at the beginning because, like I said, HIV protease is functional as a dimer. But it's also hard to do ML ensemble generation for dimers because you get a lot of structures that look like this where the symmetry is completely wrong. Um, but for these, it works very well in terms of here are the time correlation functions with the entire ensemble. And then if we filter out the ones that, like I showed before, they lie outside of the span of the microsecond MD, you know, probably not surprising, but then we get time correlation functions in better agreement with the microsecond MD. Um, so I wanted to get some more results for this before I give the talk, but then it didn't happen. But so what we're trying to work on now is do this third part here where we have instead of MD, we have some ML structures, and then we take some experimental data, and then we reweight the ML data without actually retraining the model. And so people have sort of done this before from Frank Noe's group, where you have these augmented Markov state models, where basically you have this discrete kinetic model, and then you use the experimental observables as these Lagrange multipliers to update, the equilibrium distribution and these probabilities of transitioning between these discrete states in your kinetic model. But we have a Langevin equation, so we have a continuous kinetic model where we can use the same pipeline we had before. We can estimate experimental observables like chemical shifts from the ML ensemble plus this Langevin theory. Then we take experimental data and then reweight the ensemble against the experimental data using something like a Bayesian maximum entropy approach. We recalculate these potential mean forces, and then we can iterate until the weights converge. And then we should be able to use this method to do a, more robust filtering of these structures and importance sampling and things like that. So we can actually remove structures from the ensemble that, don't agree with the experimental data, which is probably a little bit better than just looking at microsecond MD, and using that as our benchmark because at the end of the day, if the things you're calculating don't agree with experiments, whether they're ML or MD, then you need to think about what you're doing more carefully. So the conclusions is why these conformational ensembles matter. They matter because proteins must sample a conformational ensemble of states to carry out their biological functions. And even with structural biology methods like AlphaFold 2, you still need to have MD at this point, I think, because we don't trust, I think, the outputs from these methods yet. So the MD is sort of like our first step in sort of, I would say, benchmarking the physical reality of these outputs. But then these machine learning approaches, they can give us good prior structures for, um, cheap. Basically, they're much faster than doing MD. MD microsecond MD takes a day-ish for these systems. And then BioEmu, you can get the ensembles that I was showing in, maybe 15 minutes. So it's a big speed up. But they might have these deep fake structures sitting around. So acknowledgements, my current advisor, Kristen Littlefield Larson. Pratoush Tiwari was my postdoc advisor when I started this with Akash, who's a PhD student in that group still. And then I worked on the LE4PD stuff. So that paper from 2021 was when I was a PhD student at the University of Oregon, where Marina Guenzo was my advisor. So that's it. Thanks for sticking around.